Hello, everyone. My name is Cory Padilla, and I'm here from Dovetail Genomics to talk about some exciting work that we've been doing in the field of structural variation. And so what I'm going to talk to you about today is being able to capture SNVs and large structural variants in terms of inter-chromosomal translocation events using a 3D NGS approach to genomics. And so just to set the stage here, one of the things that we do know about short read whole genome sequencing approaches is they generally have a hard time in characterizing large structural variation. So this is largely in part because they're limited in the long range interactions that they are able to capture. They often require high read support to be able to correctly call breakpoints. And there's very low consensus across different tools to call these structural variation events. And here we're just showing you the different types of large structural variation that can be contained in genomes and that people are interested in characterizing. So our solution to this approach, or to be able to characterize large structural variation, is this process called HiRise, and it's really based along this technology called proximity ligation. And so just to walk you through the molecular biology, at the bottom here, starting on the left, we cross-link DNA inside of the cell. So we capture that 3D orientation of the genome. And then once that genome is kind of frozen in place, we fragment it. Historically, that's been done with restriction enzymes. We have used different approaches here at Dovetail to make that a much better approach, and we'll talk about that here in a second. Then once that genome is fragmented, we tell it to ligate back to itself, but instead of ligating back in a linear fashion, it ligates back in terms of what's closest to it in 3D space. And so what you get after that ligation and then removing the cross-links is chimeric DNA that's reflective of 3D proximal DNA rather than linear information. And then once that chimeric DNA is cleaned up, you just turn it into a sequence library where you pull down on biotin, enriching particularly for those ligation sites. Now, I mentioned in the previous slide that historically, fragmenting the genome has been done with restriction enzymes, and this generates quite a bit of problems when it comes to including all of the genome. So what I'm showing here are genomic coverage profiles on the left, and then on the right are the read pileup in the integrated genome viewer. And each one of the rows is a different approach to HiRise in terms of fragmenting the genome. So you could use a single restriction enzyme, you could use multiple restriction enzymes, or what we do here at Dovetail is use a sequence-independent endonuclease to fragment. And the dotted line in each one of those coverage profiles is shotgun. So here we're looking at how many bases exhibit 20x or 40x coverage. So the coverage is on the x-axis, number of bases is on the y-axis. And so if you look at the restriction enzyme approaches, there's a large number of bases that exhibit low to no coverage. And on the flip side of that, there are quite a number of bases that get over-sequenced. That makes sense because if you're cutting and enriching the data restriction enzyme sites, you're going to be over-sequencing those. And what that's left with is a bunch of gaps in your genomic coverage. So on the right, the restriction enzyme approaches, there's clearly large gaps in coverage. And at the very bottom row with the Omni-C, we are able to recapitulate shotgun-like coverage. And that's reflected in the read pileup, showing nice uniform signal there. So we can actually capture the SNPs, and that'll come into play later in the talk. So what this sort of increase in coverage profile plus that long-range 3D information allows us to do is expand the ability for short reads to act as long read information. So you can now do large structural variation detection. You could do your SNP and indel genotyping. You can do haplotype phasing. You can then assemble and scaffold your genomes based on those 3D contacts. And then you can also dial into the chromosome conformation, which is really the key driver, or one of the key drivers in how genes are being transcribed. So bringing regulatory elements that are up to megabases away from the gene of interest together, mediated by CTCF and cohesin to turn that gene on. And so really what we're going to do today is focus on the structural variation detection, and then we'll end with the SNP and indel genotyping capabilities and what that means for building phased diploid assemblies. And so one of the things that we want to start with is orientating you on how to read these data. So here is a sort of pictograph of how you would visualize these Hi-C data. So each one of these points on the graph is a read pair, with the x-axis being the position on the genome and the y-axis being the insert size that's captured in those read pairs. And so for each one of these dots, you can see two lines kind of running away from it as a triangle representing a region where read 1 maps to and a region where read 2 maps to some distance away. And that distance is informative of how the genome is folded in on itself. So what this really looks like is, this is a picture of two chromosomes in a genome, and we're looking at the contacts between within chromosomes and across chromosomes. And as you can see, so if you look at chromosome A, there's a lot more contacts within chromosome A than there are contacts across chromosome A to chromosome B. And the other thing is too, is that you can see there's a scale there where there's a lot of black pixels, and then there's some red pixels kind of close to the x-axis. That just indicates there is more contacts in the shorter range than there are in the longer range, just indicating that regions that are kind of close to each other in linear space have a higher frequency of contact than regions that don't interact. And so one of the things to point out here is that there is very little trans noise or inter-chromosomal interactions, as you can see with the big gap there in between chromosome A and chromosome B. And that gap is a place that we look in our contact matrices to call inter-chromosomal translocation events. Another way to view these contact matrices is through a pairwise perpendicular matrix, where you start with that sort of triangle picture, which is really only the chromosome A and B on the x-axis with the insert size on the y-axis, like I showed in the previous figure. The other way to think about it would be to make a mirror image of this to be a pairwise matrix, where chromosome A and B are on the x-axis and the y-axis, so you can actually just ask the question, how many contacts do I have between any given region in chromosome A with another given region in chromosome A, or how many interactions are there between chromosome A and chromosome B. So these are both basically depicting the same type of information. One is just pairwise, giving you that sort of square view, and one is just using a single reference genome for the triangle view that you see on the left. And so, as I was pointing out earlier, that sort of blank space in the middle is where we look to classify and detect structural variation. So what we're showing here are different contact matrices that occur within the well-characterized cell line HCC 1187. And the top row is the control BL cell line that doesn't have structural variation in it. And below that, we're showing the contact matrix for the HCC 1187 line, which has known variation. And so what we're pointing at here in every single one of these arrows is the actual inversion, deletion, or translocation event. And then at the very bottom window, we're zooming in on that structural variation, so you can see exactly where the breakpoint is and how far that breakpoint or how far that variation expands between the two chromosomes or even within a chromosome. And then you can start asking questions about the genes that are associated with these breakpoints. And so for the next part of the presentation, we're really going to be focusing in on the inter-chromosomal translocation events, kind of what we're showing here on the far right column, where we see a translocation event between chromosome 10 and chromosome 13 in HCC 1187. And next, we're going to show you how exactly do we identify and capture these variation events. So to start with, what we've been really working on in our R&D team is to develop an algorithm to be able to catch these structural variation events. So it starts by looking just at the number of chromosome pairs. So asking between chromosome 1 and chromosome 2, are there any contacts that are of higher frequency than expected by sort of the basal background level of contacts in between chromosomes? Once we have that information and identified the regions that have higher contact, we then filter out the contacts that are generated by the background noise. From there, we normalize that contact matrix to be an observed over expected ratio. And then from there, we use image processing to look at where those contacts are actually occurring across the chromosomes. And just to kind of walk you through an example of this, we're looking again at HCC 1187, and we're going to characterize a chromosome, a translocation event between chromosome 2 and 20, where we're plotting here on the x-axis the position on chromosome 2, and on the y-axis is the contact score between all other chromosomes. And so instead of plotting or having a legend for every single color for every chromosome, we're just calling out here the important contacts. So we have chromosome 19 and chromosome 20 that exhibit contacts that are over the expected rate, which is that dotted line, the horizontal line. And so then we can ask the question, okay, of chromosome 20, what does the contact matrix look like from there? And so after we filter out the sort of noise contacts in the background that are below that expected rate, we get an image that looks like this, where we see an increased number of contacts between chromosome 2, which is on the x-axis on the top, and chromosome 20, which is on the y-axis. And as you can see, there is this really bright spot between the chromosome, this particular region of chromosome 2 and chromosome 20. Now, if we remove what's, after we normalize the contact matrix for what's observed versus expected, we get a clearer view of sort of the hot spot there of the contacts between the two chromosomes. And then from there, we make it binary. So we can say the white region is the region that is of higher contacts. The darker regions are sort of background noise that we don't expect to play a role in the chromosomal translocation event. And then from there, we use image processing to then say where the actual breakpoints exist based on the contact matrix. So here we're looking at the regions of chromosome 2 in the first 3 columns there, and then the regions of chromosome 20 that interacts with chromosome 2, and then what the minimum score is, that 1.83, which is the sort of expected rate of contact between those two. And we're basically saying that this contact region, that these breakpoints exhibit a contact frequency that's higher than the expected rate, which is, again, what we see when we just look at the number of pairs that are exhibit a higher level of contacts and the sort of score figure. And so what we've done is we've tried this with a number of different cell lines at a number of different sequencing depths here. So we're looking at different cell lines between breast cancer with HCC 1187 and melanoma in colo 829. And then we also have some negative controls with the HCC 1187 BL line, the GM 12878 cell line, and then the BL cell line for the colo as well. And so even at different coverages, what we're able to do is capture all of the true positives that exist in these cell lines, and we have a very low false positive rate. And those asterisks in the false positive column indicate contacts that actually are supported. So these are translocation events that are supported by our contact matrices that have yet to be actually characterized in these cell lines. And in a bit, we'll show you exactly what those sort of false positives that we think are true positives look like. And then we're basically capturing very few false negatives with our data type as well. So this works across a range of sample types or cancer types, as well as a range of sequence depth. And to really sort of call out here the sensitivity of this assay at low sequencing depth, we've taken the same cell line HCC 1187 and we've downsampled from 46x to 6x to 3x coverage and have asked, are we still capturing the same true positive signal, and are we still not calling any false negatives? And so what we're showing here in the image on the right in the left column, we're showing the position on chromosome 14 for the x-axis, and again that contact score on the y-axis. And as you can see, as we move down between 46x, which is the top row, between 6x and 3x, which are the bottom two rows in coverage, that same signal, the same pattern of increased contacts between chromosome 14 and chromosome 20 is easily observed and is very much above our signal to noise ratio there. So indicating that we are truly calling these translocation events even as we move all the way down into 3x coverage, which is really, really, really low coverage. And then for the false negative, which is a known translocation event between chromosome 5 and 2, we can see that as we kind of downsample, we lose we maintain the contacts between 46x and 6x coverage, and then that signal kind of goes away at 3x. So we're fairly positive here that we maintain a high degree of sensitivity even as we move all the way down into 3x coverage. Now, for those translocation events that are false positives that haven't yet been characterized, but we actually have contact matrix support for them, is this one particular one in HCC 1187, which is between chromosome 4 and chromosome 8. So on these contact matrices, the one on the left is from the control line, which is the HCC 1187 BL. And we're plotting the contacts between chromosome 4 and chromosome 8 on the y-axis. And then the one next to that is the HCC 1187 cancerous line. And as you can see in the control line, there is really no bright spot here in the contact matrix. It all kind of looks like background noise. But when we look at the HCC 1187 line on the right, we see this really bright spot that pops out at us that basically indicates that the very sort of start of chromosome 4 is interacting with a region of chromosome 8 between about 15 megabases to about 45 megabases along chromosome 8. So we do have very kind of strong evidence here that this is a true structural variation that has yet to be characterized in the cell line. And our next steps here are to go in and PCR this out and just confirm that it is in fact a true signal. So I'm bringing this sort of coverage image back up here because we're about to dial into the SNP abilities of this data type as well. So previous versions of this high CN weren't that single nucleotide variants, and that because again there are gaps in coverage, and our Omni-C, as you can see again at the bottom, we're recapitulating shotgun-like coverage, meaning that we should be able to call SNPs and single nucleotide variations at a rate that is similar to that of shotgun data. And so we've done that here with the cell line GM12878. So we're looking at the table on the left, which are SNPs that were identified in the cell line GM12878 using GATK. And we have true positive, false negative, and then sensitivity and precision for the columns. And then the rows are shotgun data, the Omni-C data, and then restriction enzyme-based HiC data. And basically the key thing that we want to point out here is that the sensitivity and precision of Omni-C in being able to detect SNPs is very close to that of shotgun data, which is what we expect based on the coverage, and it far outstrips the restriction enzyme-based HiC data. Now, the table on the right is asking, can we successfully phase the haplotypes in GM12878 with the SNPs that have been identified through the Omni-C dataset, and comparing that again to shotgun data and HiC data. And so basically the punchline here is that Omni-C data that are sort of sequence independent approach to fragmenting the genome is able to capture more heterozygous SNPs than single or restriction enzyme-based HiC, which is what we expect based on the coverage. And we have a much lower switch error rate than what we see with, again, other HiC approaches. It's not as good as shotgun, but it is much better than other HiC approaches. But one of the things that we like to point out here is that you can phase chromosomes end to end with the proximity ligation data, which is something that you can't do with shotgun data because your insert size with shotgun data is about 1 kb at best. With the proximity ligation approach, your insert size is basically the range of the chromosome. And so if you have read 1 that you know contains a lot of heterozygous SNPs that maps to one part of the genome, you can take read 2 that might be megabases away and know that that particular read is a part of that same haplotype because it's linked over a long range of distance. So you can pretty much phase across the centromere, which again is something that shotgun data can't do. And then restriction enzyme-based HiC data just can't really quite get the same switch error rate because it's too gappy. There's too many regions with low coverage. And so one of the cooler things that you can do with this information is if you have PacBio data, long-read data, and you have this proximity ligation Omni-C data, you can pre-phase your long-range reads into 2 buckets and build haplotype assemblies from scratch, and which is what we've done here with GM12878, where we've taken, again, the PacBio data, aligned our Omni-C reads to them, and then have created 2 haplotype buckets of PacBio reads, and then have generated and scaffold assemblies for each haplotype de novo. And here we're just looking at sort of the genomic assembly stats here. What's the total size between the haplotypes, the N50, the N90, and the max scaffold size, realistically showing you that we are capturing the full genome in each one of these haplotypes. And then below, we're showing the link density plot, which is basically saying what is the linkage between each one of these read pairs. And you can see that these sort of hot spots that are going along the diagonal are basically the length of a chromosome. And so we're recapitulating the 2 haplotype assemblies from scratch here. So if you have a genome that's maybe exhibiting a high degree of variation, like say it has chromothripsis, you can use this approach to probably build out a new assembly from scratch of that very broken and fragmented genome. So instead of comparing it to a reference genome, you can build your own that's informative around those structural variation events. And just sort of as a proof of principle here, we've looked at how this phasing works between GM12878 and the HG002 genome. And basically what you're looking at here are k-mers that are coming from the mom on the y-axis versus k-mers that are coming from the paternal line on the x-axis. And you can basically see that we're very much able to separate out these k-mers into their specific haplotypes. So again, kind of recapitulating the idea that we can successfully build diploid assembly from scratch. So leveraging that the SNP ability along with that long-range information, we're really able to build these diploid assemblies. And one of the last things that we really want to point out here is that you can hit this data type with a hybrid capture panel. Historically, because Hi-C data use restriction enzymes, you had to really redesign your probe set to incorporate a restriction site. Because we don't rely on those, you can take off the shelf products here, like extrin lockdown and true site, or really any other hybrid capture panel, and apply it to the data. And so on the left here, what I'm showing is a workflow of how you would go from your sample to your hybrid capture approach, and then how you would process that data. And on the right, we're showing the different types of products that we know we're compatible with. And then on the bottom, we're showing one of those contact matrices that are reflective of topology. And then we're on the bottom, we're showing data that has been enriched for RPL5 using the extrin lockdown series. And I've been able to not only enrich the data for that one specific gene with the RPL5, but we're now able to ask the question, what other genomic regions does RPL5 have significant interactions with, which are depicted there by that arc? So if you kind of already know that you have a gene of interest, you can now start asking the question, what is this gene interacting with? And are there SNPs upstream or downstream of this gene, or even within that gene, that impact the connectivity between the rest of the chromosome? Or if there's a translocation event, if it interacts more frequently with a chromosome that's not where it lives. So I just wanted to kind of point that out, that you can really sort of dial in this approach to maybe not be genome-wide, but to be more gene-centric. And so we'd like to end with pointing out that this data type, this Omni-C data type, is really sort of a very flexible data type that allows you to interrogate everything from large structural variants all the way down to single nucleotide polymorphisms, and as well as gives you the ability to ask questions about the 3D architecture of your genome as well. And so with that, I'd like to say thank you and field any questions.